Really, I'd like to talk to you about our work in obesity, which is of course the commonest endocrine disorder. And you all are very familiar with the fact that obesity is a huge problem around the world. The prevalence has risen dramatically over the last 30 years, and if we just look at this very simple map of the world, we can see that defining obesity as a body mass index that is greater than 30, the prevalence is really reaching epidemic proportions in many parts of the world. And clearly these changes have occurred over a short time interval. Really only 30 years has been, we've witnessed this change over only 30 years. And clearly our environment is what's major driver of this rise in prevalence. The food we eat, the energy we expend are contributing to these changes in our weight across populations. But obesity is not actually a new disease. In fact, if we take a historical perspective, we can find that for a long time there's been evidence of obese individuals. A small number perhaps, but they have been there. So really work from across Europe, and this is one example of the Venus of Willendorf, a statue found in Austria of a Paleolithic figure depicting severe obesity. There have always been some individuals who were known to be severely obese. I hope you can see just about this frieze from a temple of Karnak near Luxor, which shows again Egyptian figures depicted with obesity. And we know from analysis of some of the mummies that in fact many of them were rather well endowed. So obesity has been with us for a long time, and in fact physicians have known this and have described obesity and its attendant medical consequences. Indeed, Hippocrates described that sudden death was much more common in the fat than in the lean. And he and other Greek physicians also described the consequences of obesity, including its effects on the endocrine axis and in particular on fertility. In fact, slightly closer to home, Galen was amongst the first to report that there were different types of obesity, and there was a form that he called immoderate, which perhaps relates to our severe obesity classification now, and another form which he was less worried about, which he called moderate obesity. Now moving forward, I think we've begun to understand a great deal more. Physicians in the 12th century, such as of course the great Arab physician and writer Avicenna, reported the association between obesity and diabetes. In fact, by tasting the urine and showing it was sugary, he was clear that people who were overweight were much more likely to develop diabetes. And of course we know that now the relative risk is incredible as BMI increases. And of course, this has been a phenomenon around the world. There are descriptions from China, from India, from Tibet even, and one of the descriptions I've shown here from Jamdatsu, showing that in fact therapies for obesity were even considered. They were somewhat unique, including massaging pea flour into the body, and also advising obese people to eat the gullet, hair, and flesh of a wolf. You could say perhaps our treatments haven't really improved greatly since that time. But actually, obesity as a medical disorder, an endocrine disorder worthy of description, assessment, and treatment, has been known for quite a long time, more than you might expect. In fact, the first monograph on obesity was published in the early 1700s by the English physician Short, who, as you can see noted here, was quite concerned about the huge amount of corpulency that was being seen in England at the time. And another English physician, Fleming, also was able to characterize these groups of obese people and recognize that, of course, perhaps they were taking in too much food, but noted that not everybody was eating to excess. So it is clear, and I think worth remembering, that whilst we're in the depths of an obesity epidemic where our environment is driving weight gain, we should not forget that there's always been a tendency for individuals to gain weight, and some individuals have had a particular tendency leading to severe obesity. So how do we try to understand the development of obesity? Well, again, to understand the pathophysiology, we need to understand the physiology. And we've known for a long time that there is a homeostatic system for regulating weight. And that system occurs in all kinds of animals and has been studied in great detail. And just shown simply on this schematic here, we can see that most organisms will maintain their usual weight. If you give them a lot of food, they will gain weight, but if you take that food away, they will undertake adaptive changes, change their energy intake and their energy expenditure to restore themselves to their usual weight. And that occurs over long periods of time in many mammals, in fact animals. What we know is that if you take an animal and you acutely overfeed it, and then you withdraw the overfeeding, it will reduce to its usual weight, and the same will occur for food restriction. Now, I've deliberately used the term usual weight here because there is no real thing such as normal weight. And what we know from animals, of course, there are some inbred strains of animals that are particularly prone to severe obesity. And when those animals are studied under experimental conditions, they too often retain a homeostatic response. So if you restrict their food, they will lose weight. And of course, if you take away the restriction, they'll gain the weight back. But their weight is on a different settling point to normal weight animals. So certainly the data from animals suggests that we have a homeostatic system for regulating weight, and we can tolerate changes in our acute energy intake and compensate by changing our energy expenditure, for example. Is this relevant for humans? Well, there are some examples really from traditional practices of overfeeding. Now, I'm referring to the Guru Walla, which is a Cameroonian festival, a traditional feast for fattening individuals, because of course fat individuals are much more attractive, generally I think, but also from the point of view of fertility. So this fattening festival has been studied in some detail from a physiological point of view. And what we see is during the Guru festival, where individuals are fed 1,000 calories in excess for over 60 days, of course, not surprisingly, individuals gain a lot of weight in a short time. And then after the fattening festival, and it's quite a costly exercise, this festival, for the tribe, they gradually will return, and only very slowly, to their previous weight. So that is evidence that acute overfeeding in humans can also be regulated. Now, of course, if we had this perfect regulatory system, we would not have the problems with obesity that we have today. And there are two main factors to take into account. So firstly, not all individuals respond to overfeeding in the same way. Some very elegant studies by Claude Bouchard in Quebec showed that in a tightly controlled experimental setting, if you overfeed identical twins, those twins will have a very similar response to overfeeding in terms of the amount of weight they gain, but will have very different responses across twin pairs. So different sets of twins respond differently to overfeeding and also to weight loss. So genetic factors will often influence how we respond to environmental exposure. In addition to that, as we've begun to understand more about the actual pathways that account for homeostasis, that control our weight, we've realized that those pathways are not perfect and, of course, change when people and animals gain weight. And increasingly, we're seeing that, particularly in humans, the energy balance system is biased and favors weight gain. So what do we know about these circuits? Well, a lot of the work has come from the study of animal models, and we now think of a homeostatic circuit that is focused on the brain, indeed on the hypothalamus, and key to regulating our weight. So we know that the hypothalamus receives signals from our fat, such as the hormone leptin, which is released by adipose tissue, is into the circulation and acts in the brain to tell us about our long-term nutrient stores. And in addition, we receive short-term satiety signals, which tell us about fullness in relation to meals, and those are neural signals and hormonal signals. Now, as I mentioned, a lot of this work has come from the study of animal models, and of course, as endocrinologists, you can all recognize these severely obese mice, and these mice were shown to have defects in the hormone leptin, in its receptor, and in one of its downstream targets, the melanocortin 4 receptor. So we know from genetic studies in mice that these are the pathways and circuits that are critical for the regulation of weight. What we now know is that once leptin reaches the brain, it triggers a really complex cascade of events, triggering specific neurons in parts of the hypothalamus that both increase and decrease our food intake. We also know quite a lot about the receptors and the peptides that are expressed in those neurons and how they ultimately project to different brain regions to exert long-term effects on energy balance. So just some key points here is that leptin is clearly one of the key peripheral signals. Insulin, of course, is another. They act on primary neurons in the arcuate nucleus of the hypothalamus. And these primary neurons then project to other hypothalamic areas, including the paraventricular nucleus, where here I've indicated the MC4 receptor, which I'll talk more about in a minute. But I think actually our understanding of this circuitry that controls weight is really at quite a basic stage. But nonetheless, it's a starting point, and it allows us to try to see if we can use this information for the understanding of human physiology. And really, because we were interested in human physiology, we needed a way in to what is a complex phenotype. And we wondered, because genetics had been such a useful tool in animals, whether we could use genetics to study human obesity. Now, of course, that seems somewhat surprising to many individuals because clearly environment is a major factor. So why should we look at genetics? But I hope that's shown you that there's always been individuals who've been genetically predisposed to severe obesity. And in fact, we know that even in an energy-rich environment, such as in Western Europe, for example, but now in many countries around the world, some people are particularly likely to gain weight. And those people are genetically susceptible to weight gain. Even if you just look at the average individual, in fact, even any two people in this room, 40 to 70% of the differences in their weight are due to genetic factors. How do we actually find those genes? Well, as you will know, a number of people are taking the approach of genome-wide association studies and trying to look for common variants, but many of those common variants have very modest effects, very small effects in terms of at an individual level. They might explain why one person is 3 kilos heavier than another at the most. In terms of trying to find genes that have a major effect, we have focused on individuals with severe obesity, trying to look for rarer variants, but ones that have a major physiological impact. And our initial study started with the identification that certain children would have severe obesity because they had mutations in the gene encoding leptin, analogous to the leptin-deficient mouse that I showed just recently. And these children developed severe obesity from a very young age. At the age of 3, this child was weighing 36 kilos, and they really very dramatic obesity that progresses throughout life. And they also associated with a number of neuroendocrine abnormalities. They fail to go through puberty, have frequent infections, and have impaired immune responses. Now, we've been able to show that we can treat these children with injections of recombinant human leptin and restore them to normal weight. Really proof of principle that the lack of leptin is the cause of the obesity in these patients. And through a series of studies over many years, we've been able to understand a great deal about how leptin is impacting on human physiology. And the most obvious thing that we noticed was that in the absence of leptin, these children are incredibly hungry and driven to eat. They're constantly hungry, they always want to eat food, and actually they'll eat any kind of food. Even English hospital food is very exciting for them, which I'm sure you can all understand is abnormal. So there's this incredible drive to eat. These children feel they're going to starve to death if they don't eat food, even though they are obese, because their brain senses a lack of leptin, which effectively makes them think they are starving. They're incredibly hungry. When we treat them with leptin, within 4 days, it's a dramatic change in their eating behavior. And when we measure this, we see an decrease in their ad libitum food intake. And over time, we of course have been treating these children for many years now. We can modulate the dose of leptin to regulate their eating behavior. Now, we returned some years later to this observation that these children not only are driven to eat, which would fit from a kind of hypothalamic understanding of the regulation of eating behavior, that physiological drive to eat, but also that they like all kinds of food. In fact, there's an incredible emotional response to food in these children. So we wanted to try and see if we could study what happens in the brain of these children when they're presented with food. And one of the tools that we can use is functional MRI, where you get not only anatomy of the brain, but also function. You see regions of the brain that are activated when patients are lying in a scanner and subjected to a particular task. And in this case, the task that they were subjected to is a very simple experiment. They were shown pictures of food versus non-food as a control. And also we recognize, of course, that not all food is the same, and I must confess that I'm slightly biased in the choice of some of these images here, but we had some slightly high-calorie foods or appetizing foods and some sort of unappetizing or rather bland foods to compare the brain response to different types of food. And what we saw is in this very simple paradigm that when the patients who are leptin deficient see any food at all, even plain cauliflower, broccoli, vegetables, they have huge activation in many areas of the reward centers of the brain. There's many of the areas that are triggered by nicotine, by alcohol, by drugs of abuse, are highly activated by food in these children. When we treat them with leptin, that response is attenuated, goes down within about 7 days. Even when they haven't lost any weight, there's an acute effect of leptin on that brain activation response. And this response in the brain mirrors changes in their behavior. So in the absence of leptin, they like any food. After 7 days of leptin treatment, they will change that behavior, and now they will just go for the chocolate and they will turn away the vegetables like any normal person would want to do. So an acute change in the brain and in behavior after giving leptin, showing us that these very complex behaviors are modulated by physiological signals. And really just to sort of drive home the value of this is that albeit a rare disorder, it is really life-saving treatment for children. Here are two of our most recent patients that we've treated, referred by colleagues in Turkey. And now we're able to treat children as young as 1 year old and really restore them to normal weight. And these children will progress through puberty at an appropriate developmental stage. When we do pulsatility studies, we show that they maintain flat pulsatility in the prepubertal stage, and then presumably in response to other developmental cues, will go through puberty at the appropriate time. And now we have some young adults who are treated, who have normal reproductive organs and we think will appear to be fertile. And just really finally show you here that these children, in fact, these are two of our leptin deficient patients shown in Egypt, really are entirely normal and can live normal lives once they're treated with recombinant human leptin. And we're really very grateful to Amgen and Amelin, who've continued to provide us with recombinant human leptin free of charge for the support of these patients. So moving on from leptin, we've now been able to show that mutations in many of those hypothalamic pathways identified in mice cause human disease. And just I've represented the work of many people in the team shown here, where we've been able to find mutations in several of these molecules in association with severe obesity. Now, of course, finding the mutation tells us that these pathways are relevant for human disease, but it also allows us to study groups of these patients to try to understand the physiological consequence of mutations. And we've learned a number of key things. So firstly, we've noticed that all the genetic forms of obesity that we've described to date are associated with hyperphagia, the drive to eat. Just showing some examples here from the commonest genetic form of obesity, which involves mutations in the melanocortin-4 receptor, we can show that all of these children have an increased food intake, they're driven to eat, but also when we compare this very large group of individuals by the severity of their mutation, we find that those who have a more severe mutation eat more than those who have a milder mutation, showing how critical it is to have the right degree of melanocortin tone in terms of modulating eating behavior. And we also learn quite a lot from these patients that sometimes reminds us that we can make some assumptions in clinical science. So for example, we were keen to look at how brain reward may be altered in MC4 deficiency. We knew that the leptin patients have heightened reward to food. Now, when we did the same brain imaging study showing pictures of food and non-food to patients with MC4 mutations, we found that actually the striatum was activated to a comparable degree. It wasn't disordered. But where the difference actually lies in that response in MC4R obese patients is very different to individuals with common obesity who we studied as our controls. Now, this is slightly complex, but I think what it means is that in common obesity, something goes wrong in terms of the brain response to food. When regular obese people see pictures of food, the appetizing food, the burger, the cake, their striatum, the reward areas, should light up, but it's not lighting up. This has been hinted at by some other studies, but here in this study we see a clear difference between the two obese groups. And we think what this means is there are some changes in the neural circuitry that occur with common obesity, with weight gain. And those changes in the neural circuitry need the melanocortin-4 receptor. So when the melanocortin-4 receptor is not working in the group of patients that we are studying, you don't see those conventional changes. So there's quite a lot more we need to understand about how this complex circuitry interacts. We've also been able to show that this leptin-melanocortin system is critical in linking nutritional signals with the endocrine axis. Of course, we've known for a long time that changes in weight affect fertility, and of course I mentioned leptin is a key modulator of the reproductive axis. But we also, of course, know that changes in nutrition affect growth. We'll be very familiar with the fact that children who are deprived of nutrients at critical stages in their growth will become short, and if you give a lot of nutrition, people will gain height and become taller. One clue to that may come from the study of these patients with MC4 mutations. Here we find that the patients are very tall. Now, we know that obese people are taller in general, obese children are taller, but even if we match for age and for the same degree of obesity, patients with MC4R mutations are much taller than we'd expect. So what is driving this accelerated linear growth? Well, one feature may be the fact that they have high levels of plasma insulin, again, more than we expect for the degree of obesity. But another interesting feature was shown here when we undertook some pulsatility studies to look at growth hormone pulses in adults with MC4 mutations. Now, you will know that in common obesity, in fact, as people gain weight, their growth hormone pulses become flat. That's the conventional response with weight gain and obesity. Yet here we see that obese individuals, identical BMI to the controls, retain pulsatile growth hormone secretion. In fact, the area under the curve is about 70% that of lean individuals. So we think what's happening is that there is incomplete suppression of growth hormone pulses, suggesting that melanocortin 4 receptors impact on somatostatinergic tone. So the other thing that we've been looking at is this common clinical paradigm that many of you will be familiar with, is that patients will tell us, look, I'm not really eating that much, doctor, but I'm still putting on weight. And the response of most physicians is, well, you must be lying because you can't really put on weight if you're not eating that much. But perhaps, rather than jump to that conclusion, we should try to undertake some measurements. Well, of course, we know that in the MC4R deficient patients, they do eat a lot and they are hungry. But we've been keen to try and measure their energy expenditure, looking for deficits that certainly the mouse studies would suggest we should find. But in fact, whatever we do to look for defects in energy expenditure, we don't find any at all. So no difference in basal metabolic rate, free living energy expenditure, chamber calorimetry, any tool that we use, we cannot document differences in energy expenditure in humans. However, what we do see is when we undertake indirect calorimetry, we find an increased respiratory quotient in patients with MC4R mutations. And this is the ratio of carbohydrate to fat oxidation. And we think what is happening here is that the MC4R deficient patients have an inability or reduced ability to oxidize dietary fat. Now, why should that happen? Why should you not burn calories as fat? Well, one clue again comes from the mice, and the mice have impaired sympathetic tone and impaired lipolysis, and therefore release much less fat. We think something similar may happen in humans. We do have a clue that humans with MC4 mutations have impaired sympathetic nervous system activity by a phenotype that is actually more easy to measure, and that is blood pressure. And this is work done by Jerry Greenfield, who is a fellow working with us and has now returned to his native Sydney. And Jerry, working with Julia and Lana and my team, were able to show that patients with MC4R mutations have a much lower prevalence of hypertension than you'd predict for the degree of obesity. In fact, their average blood pressures are about 122/72. Very good blood pressures for people with lifelong severe obesity and a mean BMI of 45. So why are they relatively protected from hypertension? Well, it could have been differences in insulin secretion and sensitivity, but we undertook clamps and there's absolutely no difference between MC4 patients and controls. However, during the clamps, Jerry had the idea that we should measure heart rate and heart rate variability. And what we see is that when the patients are sleeping, their heart rate is the same as obese controls. And of course, heart rate when you're sleeping is mostly due to vagal tone. When they wake, their heart rate increases, as it does in obese controls, but it increases to a much lesser degree. And then when, of course, the intravenous insulin is turned on, there's a further increase in heart rate. But again, the magnitude of that increase is a lot less. And right the way through the duration of the clamp, and even here at steady state, the heart rate is 8 beats per minute less. And this data, plus additional data on urinary catecholamines, would suggest that these patients have impaired sympathetic tone, and therefore that this melanocortin system is a key link between weight and blood pressure. So in addition to doing the physiological studies, we've done a lot of studies in the lab looking at how the mutations impact on the particular molecules. Now, the MC4 receptor is a G-protein coupled receptor, and we've studied a lot of the mutations in cells and modeled their impact on the receptor, and show that many of the mutations are missense mutations that prevent the receptor from getting to the cell surface. Now, for us, the main reason to understand the molecular mechanism is, can we use that information to develop more targeted therapies? So here in some work we've undertaken collaboratively with Michel Bouvier in Montreal and colleagues at Amicus Therapeutics, we were able to see if we could rescue the cell surface expression of some of these mutated MC4 receptors with various drugs. So the arrow here indicates the number of wild-type receptors at the surface of a cell. Here we're studying a particular mutation found in patients, and you can see a reduced cell surface expression. And then over here, we're adding a pharmacological chaperone, which in cells increases the number of receptors at the cell surface. And because this works for some drugs and not for others, and for some mutations and not for others, we've then been able to model how the drugs interact with the receptor to improve its function and binding. And really, it's this kind of work and others that is leading to the development of drugs working on this pathway. There is now a potential drug which does not have the predicted effects on blood pressure that we were worried about, but could be used as a potential therapy in patients with MC4R mutations. So how far have we got? Well, I mentioned to you that we've been studying patients with severe obesity, and really all our work has been based on this cohort of patients that we've recruited over many years to the Genetics of Obesity Study, or GOOS. And really, with the help of many colleagues in the UK and indeed worldwide, we now have a cohort of over 4,500 patients with severe obesity. The genes that I've told you about already have shown us that these pathways involved in animals are important in humans, but interestingly, these mutations collectively only explain 8% of the obesity in the GOOS cohort. And we're pretty convinced that there's a lot more to find, and that's really based on the patients. You can see these children here on the left of the slide. They're severely obese at a very young age. There is clearly something going wrong that is causing this level of obesity. And really our aim now is to try to find out the additional genes that we think could explain these phenotypes. And we're taking two main approaches. So firstly, we continue to get clues from animal models and look at new candidate genes. But really, as our knowledge of the human genome has improved and the technologies have advanced, we can now take completely hypothesis-free approaches to finding genes. We don't have to know the gene or have a clue about it, but we can try and find the genes that might be causing the obesity in the patients. One example of that work is shown here, where we've looked at copy number variants, so deletions or duplications on the chromosomes which take out certain genes and might explain obesity in certain patients. This is a bit like Prader-Willi syndrome, where you'll be familiar with deletions on the imprinted region of chromosome 15. Here we looked at 1,500 GOOS patients compared to almost 8,000 controls, and we showed that a lot of these patients have copy number variants, but that each variant is very rare. And I think this is an emerging theme in obesity, is there will be many genes, but only a small number of people will be affected by each gene. So here's one particular deletion we focused on, and that involves chromosome 16, where we found a small deletion that takes out a number of genes, including SH2B1, which is involved in leptin and insulin signaling, and indeed in growth hormone signaling. And we've gone on to show that the patients who have this deletion are severely obese, but also have very high levels of plasma insulin, greater than we'd expect for the degree of obesity and consistent with disproportionate insulin resistance. They also have a reduced final height. So we have some clues already. We have clues from genome-wide association studies with multiple common variants. I've shown you quite a few of the rare variants that we've identified that lead to a more severe phenotype. But we still have a lot of missing heritability. A lot of the genetics of obesity remains unexplained. Do we think that there are more genes to find? Well, of course, there are several explanations for this missing heritability. There may be epigenetic effects. Of course, the environment is a major driver impacting on genes. But we think there are more genes to find, and that's because the heritability of body weight is very similar across species, regardless of the environmental factors. The way that we think we can find some of these missing genes is really using some of the emerging technologies, including whole exome sequencing. And this is just a very simple schematic shown here. But what we can now do is, rather than sequence one gene at a time, we can sequence all of the protein coding genes in the genome in a single experiment. So this is very exciting, and it gives us a huge amount of data. Of course, the challenge is, how do you get to the real cause when you have this huge amount of data? And I think that's a challenge for all of those working in the mechanisms of endocrine and other diseases. Just to give you an example, on average, from this kind of data, you get 40 to 50,000 genetic variants. If you take out all of the variants that are found in controls on various databases that are known, you still often end up with 5,000 genetic variants that are unique to the patient in front of you. And that's really a much bigger number than we realized. So we tried to restrict it a bit further, and we focus only on those variants that results in changes in amino acids, where we can more readily, we hope, tie in a loss of function to the phenotype. But even then, we have often about 500 or 600 variants to look at. And really, our I'm going to show you a couple of approaches that we're taking to try to link these variants to disease. So one approach we can take is to use traditional Mendelian genetics. So we have a number of families that are consanguineous, where we have recessively inherited severe obesity. And here we can use whole exome sequencing to narrow down the regions of interest. So one example shown here, there are 3 individuals who share very large blocks of homozygosity which contain 163 genes. How do we find the gene amongst them? Well, we look for any obvious candidates. We didn't find anything. We add in whole exome sequencing, and we can very quickly narrow down to only variants in homozygous regions that are shared amongst the affected individuals in a family. Another approach that we can take is really almost back to basics in terms of clinical science. Think about the patient. Think about the patient with the unusual phenotype and try and work out what might be explaining it. Well, within our GOOS cohort, we have many patients who have not only obesity but some other features. So, for example, some will have clear evidence of hypothalamic disruption, salt-water balance, some neuroendocrine abnormalities. Sometimes they can be very subtle, and certainly patients report disorders of thermoregulation, of sweating, etc. We have a number of patients, quite a large group, who have a range of behavioural abnormalities, again, more than you'd expect by chance alone. And a number of patients who have various developmental abnormalities. So one of the tools that we've developed is a bioinformatics tool which allows us to plug in a list of genes from whole exome sequencing and filter them by phenotypes of interest and in association with obesity. And using this tool called Gander, we recently collaborated with Chris Chatterjee, endocrinologist in Cambridge, who many of you will know, to identify a patient with a thyroid hormone receptor alpha mutation who had a phenotype that included short stature, obesity, and abnormal thyroid function tests. By plugging those into our bioinformatics tool, we were very rapidly able to come to the potential pathogenic variant in this patient. But really the major part of our work right now is focusing on those patients where we don't have any other clue and yet they have severe obesity. And as part of the UK 10K consortium, a major consortium in the UK funded by the Wellcome Trust, we're able to undertake whole exome sequencing in 1,000 UK children with severe obesity. And this is just a very simple outline of our approach to the analysis, which at the moment is focusing on, after excluding the known human obesity genes, on tackling the genes which we know cause obesity in mice, and first of all trying to see if we might find some human mutations. And we certainly are following a lot of this work up in families and then further association studies and functional studies. But our early data is certainly suggesting that there are many, many genes, in fact many more than we even estimated, being involved in human obesity. And of course, I want to show you a little bit of just how we propose to take forward some of the genes in terms of investigating their impacts on function and physiology. I mentioned to you, of course, we do a lot of work in cellular systems, but in addition, we wanted to work up a system for studying the impact of these genes on the brain. And one way that we can try to do that in humans is using inducible pluripotent stem cells, which can be used to derive neurons in culture. So just for those of you who may not be familiar with the work, you can actually take a skin biopsy from a patient with a genetic form of obesity, take those fibroblasts and reprogram them into stem cells, and then take those stem cells and differentiate them, if you wanted to look at the hepatocytes or pancreatic cells, but of course for us, we're interested in neurons. And doing that work, we're now developing neurons in culture which have come from those fibroblasts and stem cells. And those neurons carry the genetic abnormality of the patient from whom we took the biopsy. And this is really very exciting work. There are quite a lot of challenges ahead, but we can show that these neurons, for example, express the leptin receptor, they respond to leptin, and we can show that they can fire, and also we can look at their synaptic interactions. In addition to looking in culture, of course, ultimately, these genes are acting on physiology and on behavior. And we have we're growing a program of work looking at eating behavior in a lot more detail, trying to really understand the different components that determine what we eat. So, of course, as an endocrinologist, I've always been interested in those fundamental drives to eat and in satiety. But of course, we recognize that things like taste, the liking of food, and higher cognitive effects, stress, emotion, motivation, all determine what we eat. And it's really integrating that information to try to find a model with which we can then map on the genetic and molecular mechanisms. So really, I just want to finish by saying that I hope I've shown you that we can demonstrate that rare, highly penetrant genetic defects can lead to severe childhood onset obesity. And through the study of these genetic disorders in these patients, we've learned a great deal about the molecular and physiological studies, mechanisms involved in the regulation of weight, which show us that these are highly conserved across species. It also reminds us, which I think is something that Galen reported and noted, and I'm sure many others before us have talked about, is that obesity is not a single entity. There are many different types of obesity. These are incredibly heterogeneous disorders. Our forefathers recognized this, and I think now in the molecular era, we may be able to try and define those in a more concrete manner. And ultimately, obesity is about the brain. And if we really want to try and understand obesity and help our patients, it'll be about understanding these neural mechanisms. So I just want to finish really by thanking many of the people who've done the work, including all of my team shown here, but in particular two very important people who've worked with me for many, many years. Julia Keogh, who's worked with me on setting up and running the GOOS study for a very long time, which has really been the resource upon which all of our work is based. And Julia and Alana have together undertaken all of the clinical studies I've shown to you today with me and the treatment of the patients with leptin deficiency from around the world. So really, I'm incredibly grateful to their hard work and their dedication that really has enabled this work to happen. I just want to acknowledge my collaborators in Cambridge in particular, but also our many collaborators across the world with whom we couldn't do this work, which is indeed highly collaborative, and our funding bodies shown here. Of course, this is a prize for a relatively young endocrinologist. I was really grateful to Hans for mentioning the age limit for this award. But young endocrinologists cannot really grow and develop without great mentors. And I've been very grateful in my career to have a number of mentors who've really taught me many things. Firstly, Gareth Beavers from Birmingham, who was, if you like, almost the classic Renaissance man, if you like, an absolute eccentric, a British eccentric, who taught me that really there are no boundaries and you should just ask questions and you should take your clinical observations and then try and work out what's going wrong. In fact, he was quite unconventional, which I must say I quite enjoyed. And you can see perhaps the other two gentlemen on here are also quite unconventional. John Wass, who from Oxford, really gave me the passion for endocrinology and really was responsible for my decision to pursue endocrinology as a career. And of course, at the bottom of the slide, my great mentor in Cambridge, Stephen O'Reilly, who really is the clinician scientist par excellence, who I think, as well as persuading me that obesity was a very important disorder, often neglected disorder, but worthy of scientific study, also I think as a role model has shown me that you can be equally comfortable studying a molecule as you can talking to a patient about what they eat and how much insulin they take. And I think to be equally good and competent at both of those is an incredible challenge, and I'm very grateful to my mentors for having guided me. And finally, really the most important people of all are our patients. And they're the reason that we do the work that we do. They give us the ideas, they contribute by volunteering themselves to partake in our research, and really it reminds us that we do the research certainly for the benefit of our patients. This little girl shown here is one of our patients with an MC4R mutation, who we first identified at the age of 4, and now at the age of 10, she's doing pretty well. But I think it also reminded me to say that even when we may not be able to find a treatment just yet for the patients, the knowledge that we have an understanding of their medical condition is incredibly important. The information in this child that she has an MC4 gene defect has empowered her to actually be comfortable with the fact that she has a weight problem, so much so that she's in fact quite an advocate for this. In fact, as you can see, is fundraising for our research. She did run a cake sale, which I think is quite telling, but she enjoyed the cake sale and she raised a lot of money and she wants to use it for research into the genetics of obesity. And I think that's really, if anything, the greatest testament that we can ask for. And then finally, I just want to direct us to a website that we've recently established where you can access information about our research, but importantly, where we hope to provide information for patients, because really it's by collaborating together that we'll be able to make some developments in obesity. And really, I'd like to thank you once again.